So, welcome back to the next second session of the lecture series of protein and structural contents. So, now I will be talking to you today in this set of information about non-covalent bonds. So, if you look at a system, then a chemical composition of any biological system mainly consists of these small molecules like the amino acids, nucleotides, sugars, macromolecules, DNA, RNA, proteins, etc. Whereas, if you look at the molecular biology, which is study of higher level of molecular organization or interaction between these various small as well as large or macromolecules. So, all kinds of interactions do happen inside the cell or any organism, and that is what the molecular biology essentially deals with. Now, if you look at the different kinds of bonds that are formed because of interactions, one is definitely covalent bond, which is the classical nature which you have probably learned, which happens because of sharing of electrons. So, if you look at these atoms, for example, hydrogen, carbon, nitrogen, phosphorus, sulfur and oxygen, all of them have certain number of electrons and they can form covalent bonds by sharing these electrons with others. These electrons remain within remain with the atoms in the form of these orbitals, that is the electrons are present in any one of these places. So, and the shape of the orbitals are defined by some alphabets like the S, P, D and F orbitals, and you must be aware of these things very much. So, if you look at how these sharing of electrons they happen, they readily form covalent bonds with other atoms, and they rarely exist in nature as isolated free entities. For example, hydrogen as such, carbon, nitrogen, etc., does not remain. Well, carbon do remain, but then in different forms. So, carbon has 4 electrons. So, it completes its octet, as you know very well, and they form by complying with the octet rule, they form 4 covalent bonds. Nitrogen also can form 4 covalent bonds, and there is one pair of electron which we call as the lone pair that does not gets involved in covalent bond formation. And in ammonium, for example, nitrogen forms 4 covalent bond. Phosphorus can also form 5 covalent bonds, for example, in H3PO4. So, this is how it can form the 5 covalent bonds. So, these covalent bonds are very stable, and the energies required to break or rearrange them are much higher than the thermal energy available at room temperature. So, this covalent bond, even if you heat it, let us say at 100 degree, it is very difficult to break these, because the energy of these covalent bonds are very high. However, for if you just look at the different types of covalent bonds and what are their bond energies. So, these are the different types of covalent bonds. So, for example, C can bond with nitrogen with 3 covalent bonds. The energy here is enormous, 213 kilocalorie per mole. The primary covalent bonds, that is the CC bond, is also about 83 kilocalories per mole, or for that matter CH covalent bond is about 99 kilocalories per mole. That is, for making these bonds or for breaking up these bonds, you need to either spend this much of energy or somehow provide this much of energy for formation of these bonds. So, the covalent bonds have characteristic geometry. As you can see carbon bonds with hydrogen in this tetrahedral structural geometry, and if you look at ethylene, then you see CH2 and CH2 are bound together by this double bond, and this is more of like a planar molecule. So, depending on, if you look at this table, which is the our normal table. So, you can see that the electronegativity series in this table begins with the fluorine being the most electronegative. So, in this periodic table, the colors represent the electronegativity. As you can see, there is nitrogen, oxygen, chloride, chlorine and bromine. So, they have certain electronegativity than the yellow ones, which shows about 2 to 2.9 and so on. So, this electronegativity series also decides the covalency of covalent bond. So, the covalent bonds are therefore divided into two types called the polar covalent bond, where the electronegative atoms like fluorine forms the covalent bonds, and then another polar covalent bond is the OH bond, oxygen being again very electronegative, and it forms covalent bond with water. But then these are polar. Why? Because there is a polarity, that means the electron pair, although they are shared, are pulled towards one of the more electronegative atom more than the other. For example, in the case of OH, the O draws or pulls this pair of electron which is involved in the bond formation towards itself. Similarly with the fluorine, and then we have the non-polar covalent bonds, for example, CC, CH, etc. These non-polar covalent bonds are non-polar because there is no discrete preference for the electrons to be shared between these two atoms. So, both the atoms carbon, 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 hydrogen are essentially sharing them equivalently. So, those are non-polar covalent bonds. Now, let us look at these are the covalent bonds. However, in most of our biological systems, we require not only the covalent bonds, but a large chunk of all the interactions that happen are due to non-covalent bonds. So, what are these? As you can see, non-covalent bonds are critical in maintaining the three-dimensional structure of large molecules. So, if you look at this nucleic acid or protein, here are certain examples. So, here is some DNA, some Ras protein, etc. Each one of them are actually held together, the main chain is held together by covalent bonds, but the two chains or the protein by itself, as I have been telling you in my previous class, are held together by large number of non-covalent bonds. Each one of these non-covalent bond is weak and transient in nature, but then because there are so many, because there are multiple non-covalent bonds, they often act together to produce a highly stable and very, very specific association between different parts of a large molecule or between different molecules. For example, a protein can interact with DNA. So, those are because of various kinds of non-covalent bonds, but not one or two. Each one of these one or two non-covalent bonds may be very, very low in their energy, but then it forms so many of them that the total energy of these interactions is enormous. So, for example, the energy released in the formation of non-covalent bond is only about 1 to 5 kilocalorie per mole, which is less than the bond energies of a covalent bond like CC bond, which I just now showed in my previous slide, as about 83 kilocalories per mole. So, you can imagine it is much lesser, minuscule amount of energy in each non-covalent bond as compared to the covalent bond, but because these non-covalent bonds are formed in so much number that they are overwhelming. So, because the average kinetic energy of molecules at room temperature is only about 0.6 kilocalorie per mole, and you can see the average bond energy for non-covalent bond is about 1 to 5 kilocalories per mole. So, 0.6 is very close to about 1 kilocalories, and a few of them or a slightly higher temperature, not room temperature, but just above that temperature, the energy increases, and therefore, many of these non-covalent bonds can be simply broken. So, because of such transient nature, they are not called bonds, rather they are much better referred to as interactions. And as you can see, here are two protein molecules, and these protein molecules are interacting with each other by formation of various kinds of non-covalent interactions. Proceeding further, so what are the different kinds of non-covalent interactions? So, the first one in the series, there are 4 different kinds of non-covalent interactions. One is ionic interaction, hydrogen bonding interaction, hydrophobic interaction, and van der Waals interaction. So, I will walk you through each one of these interactions one by one. So, if you look at ionic interactions, so just now we looked at in something called the covalent interactions, where there is sharing of electrons. But in ionic interactions, what happens in some compounds, the bonded atoms are so different in electronegativity that the bonding electrons are never shared. So, one atom obviously takes away the pair, takes away electrons towards itself, because it is simply very electronegative as compared to the other one. One classic example is NaCl, you can see here. So, chlorine, which has 7 electrons, is negatively charged, whereas sodium, which has 1 electron, is positively charged. So, what it does is it just goes closer to the chlorine, and then it takes electrons, and then they form what is called as sodium chloride. So, the ionic bond or interaction or ionic interaction results from the attraction of a positively charged ion, which is in this case sodium or magnesium or calcium, or a cation, for a negatively charged ion, which is called an anion. For example, in this case it could be chlorine or oxygen, or chlorine again for this calcium. So, that is what happens, and unlike covalent or hydrogen bonds, the hydrogen bond I will be talking to you later, ionic bonds do not have fixed or specific geometric orientation, because what happens is, when we talk about a positively charged ion, the electron is essentially not situated just here, it is actually all around the sodium atom, and therefore, it or for that matter the chlorine atom, chloride ion, the electrons are all around, and therefore, it has no directionality anywhere the sodium can come and bind. So, it is uniformly distributed across the atom. Now, once they have formed these kind of non-covalent or ionic interactions, then what they do is they actually pack themselves into crystals. So, crystals of salt such as NaCl do have very regular structures, because that is energetically most favorable way of packing. The force that stabilizes such crystals is called the lattice energy. Now, what happens in aqueous solution? Simple ions such as sodium, magnesium, calcium, potassium, chloride, etc., they do not exist in free. They are always, because of the charged entities, surrounded by water molecule, and whenever they have to interact with any other, for example, sodium have to interact with chloride, the sodium ions need to shed off the water molecule surrounding it, and then it can interact with the chloride. So, if you look at the characteristics of these ionic interactions, so most ionic compounds are quite soluble in water, because as I said, a large amount of energy is released when ions tightly bind water molecule. This is known as energy of hydration. So, if you look at these two atoms, so chloride is surrounded by water, sodium is surrounded by water, but then when they interact with each other, the hydration shell is released, they form a strong ionic interaction. So, the oppositely charged ions, which are shielded from one another by water and tend not to recombine, now recombines, and once they recombine, they do not again remain in this fashion, but they immediately form what is called as a crystal lattice or a crystal. And once they crystallize, because this is the most energetically favorable state, they would like to remain in this fashion. However, if you put water in here, these crystals will immediately dissolve, because the forces of interaction of water molecule to individual ions is much stronger than the forces of interaction between one positively charged ion and another negatively charged ion. So, interaction between this and that, which resulted in the formation of this crystal, is lesser than the interaction between this and water molecule, and as a result of which, as soon as you add water to the lattice, immediately this crystal lattice dissolves. This is what happens in case of salts like sodium chloride, where the energy of hydration is greater than the lattice energy. However, some other salts, for example, calcium phosphate, are virtually insoluble in water, because the large charges of calcium and phosphate ions generate a formidable lattice energy. The energy within the packed structure is much higher than the hydration energy, and therefore, calcium phosphate do not dissolve. Given that, we will now proceed to different kind of non-covalent bonds, which is the hydrogen bond. Now, this is one of the most important bond or non-covalent bond that exists in the universe, simply because we in the universe at least Earth planet, we have 70 to 80% water. All the organisms, they have 70 to 80%, our cells have 70 to 80% water, and this water remains in the liquid form because there is something called hydrogen bond. So, polar molecules such as water, they have a weak partial negative charge at one region of the molecule. If you look at this structure here, this is water H2O. You see, at one part there is a partial negative charge, which is oxygen. Oxygen is electronegative, therefore, it has a partial negative charge represented by delta negative. So, if this has a partial negative charge, obviously, the hydrogen which is connected to this oxygen by a covalent bond is having a partial positive charge. But this partial positive and partial negative charge has caused some kind of a polarity in this water molecule. That is why they are called polar molecules, because there is a charge difference, distribution of charge, one is negative, one is positive. So, as a result of which this hydrogen, which is covalently linked to this oxygen, now this positively charged hydrogen is capable of interacting with another negatively charged oxygen. Or we can conversely place it that this negatively charged oxygen here is now capable of interacting or forming what is called as the hydrogen bond with the hydrogen which is slightly positively charged from another water molecule. So, you have one water molecule, another water molecule, and the oxygen is capable of interacting with hydrogen, and if we just increase that in 3 dimension, you can see every oxygen in any water molecule is capable of interacting with hydrogen of the neighboring water molecule, and that water molecule in turn is capable of interacting with another water molecule through hydrogen and oxygen, and hydrogen and oxygen, and hydrogen and oxygen, and so on. So, there are large number of what is called as hydrogen bonds that are being formed. These hydrogen bonds keep on forming and breaking because each hydrogen bond by itself is very, very weak. So, in these situations, a hydrogen atom, and that is why it is called hydrogen bond, a hydrogen atom which is covalently bonded to a donor atom, let us say D here. So, this is a donor atom. So, here we call it D negative, this is delta negative. So, D, and to this a hydrogen is covalently bonded, may form an additional weak association with an acceptor which is A negative. Now, here the acceptor is a hydrogen again from another water molecule, and then they form what is called as a hydrogen bond represented by these dotted lines. So, in order for the hydrogen bond to form, the donor atom must be electronegative, as is shown in this case. So, the oxygen is electronegative in water. If you look at any of these interactions where some amino acid, for example, cysteine is interacting with threonine. So, cysteine has these S, this is electronegative interacting with hydrogen, which is again interacting with oxygen, again covalently bound with oxygen. So, the oxygen is electronegative, this is the donor, this is electronegative, and hydrogen is bound to another electronegative. So, in order to form a hydrogen bond to form, the donor atom must be electronegative. So, here it is oxygen which is electronegative, so that the covalent DH bond is polar and the acceptor is also electronegative, as is shown in this particular case. Now, what happens in biological systems is both the donors and acceptors are usually nitrogen or oxygen, or those groups containing the amino or hydroxyl group. For example, methanol, ethanol, propanol, they have these hydroxyl groups, and therefore, each one of these hydroxyl group having the hydrogen bound to oxygen is capable of forming hydrogen bonds with water. So is it in the case of DNA. As you can see, there is a thymine, there is a adenine. So, these are A, T, G, C base pairs, and they have the oxygen, okay, the NH2. So, this NH can form hydrogen bond with this oxygen, this NH can form hydrogen bond with this NH, and so on. So, these hydrogen bonds are so much important in biological systems. In the last class, I was talking to you about peptide bonds and ester bonds. So, you can see in the peptide bonds also, the NH is or OH is capable of forming hydrogen bonds. By contrast, if you look at a CH or CC bond, they can never form hydrogen bonds. Particularly in CH bond, it is not polar. It will be polar only when one of the atom to which the hydrogen is bound is electronegative. So, that is what a hydrogen bond is. What are the characteristics of these hydrogen bonds? Most hydrogen bonds are very, very long. So, long in terms of covalent bonds. So, while covalent bonds are short, hydrogen bonds are long. They are essentially almost like twice the length of a covalent bond. So, here the solid line represents a covalent bond and the dotted line represents the hydrogen bond. The solid line is shorter, this one is longer, and the energy of these is about 5 kilocalories, whereas the energy of a covalent bond is about 110 kilocalories. Look at the difference. This is way higher, about 51 times higher than this bond energy. Not only that, another important feature of hydrogen bond is the directionality. So, when I say hydrogen bond energies varies from 3 to 5 kilocalories or 3 to 8 kilocalories, what it means is when the hydrogen bond is formed in a linear fashion, that means this ROH or HOH, whatever you consider the water molecule, and the next electronegative atom to which the hydrogen is attached forming this hydrogen bond are collinear, that is in the same line, then they form a strong hydrogen bond. If however, these two are not linear, that means the hydrogen bond is formed at an angle to the next acceptor group oxygen, then this angle actually reduces the strength of the hydrogen bond. So, the hydrogen bond strength in proteins and nucleic acids are only about 1 to 2 kilocalories per mole, which is much less than water. In water it is about 5 kilocalories per mole. So, the directionality also matters, because when you look at two proteins interacting, the proteins might come in various different orientation and start interacting, but they can interact by formation of hydrogen bonds, by interacting with the peptide bond, by interacting with the side chains, by interacting with any other hydroxyls or amino group available on the polypeptide. So, that kind of interaction happens at various angles and the energies are much lesser. Now, we move on to another kind of very important kind of interaction called the van der Waals interaction. This is another non-covalent interaction. So, van der Waals interaction essentially exists everywhere. For example, now I am standing here or whatever a pen is here, this is getting attracted or getting access to interact with anything on this table or this laptop or anywhere or to me. So, I can interact with anybody, another person or another non living object can interact with anything because of van der Waals interaction, and these interactions are happening every time. Actually, these are atoms that are interacting with each other. So, what happens is when two atoms, for example, atom A, look at this picture here. So, you have atom A and then you have atom B. Now, atom A, as you know, the electron cloud is surrounding this atom, but then momentarily it might happen just because of anything that momentarily large number of electrons are towards this side of the atom or this hemisphere of the atom as compared to this hemisphere of the atom. Therefore, this attains a partial negative charge and this portion attains a partial positive charge. Now, let us assume that at such an instance where this portion of the atom is highly electronegative or polar, negatively polarized, and this portion is positively polarized, has come in close proximity to another atom called B. So, you have a polarized atom and it has come in close proximity with B, it will influence an instantaneous dipole to be created on B, because there is less number of electrons, therefore, the B will be pushed in such a fashion that more and more electrons will come in close proximity to this side, because this is positively charged. So, they will come and start creating a negative charge as is shown in this figure. So, you have a partial hemisphere of negatively charged electrons, and here also the partial, because this has come in close proximity, or this is positively charged, this is negatively charged. So, they would like to attract each other. So, these kinds of interaction by influence of each other, as a result of which there is a dipole interaction and they try to come closer to each other, is called the van der Waals interaction. And as I said, these are non-specific, because I can get interacted with the table, I am actually getting interacted with the table, this pen is getting interacted with the laptop, anything is interacting with anything else, because this essentially consists of the atoms, and each atom under the influence of something else, or even the atoms that are the ions, or the atoms that are floating in air, are interacting with each other through these van der Waals interactions. So, these are very, very non-specific, anything can interact with anything else, and they are momentary and they just keep on transferring. So, if two non-covalently bonded atoms are close enough together, the transient dipole in one atom will perturb the electric cloud of the other, or the electron cloud of the other, and this perturbation generates a transient dipole in the second atom, and that two dipoles will attract each other weakly, and this weak interaction is called the van der Waals interaction. So, the strength of this van der Waals interaction decreases rapidly with increasing distance. So, until unless these two atoms that I have just now shown comes in close proximity with each other, this atom cannot influence this atom. So, as you increase the distance between these atoms, the van der Waals interaction forces decreases rapidly. So, these non-covalent interactions can only form when the two atoms are quite close to one another. But imagine the atoms have come close to each other, the one atom electron cloud has influenced the other atom, and therefore, there is a negative charge, there is a positive charge, they try to attract and they form some kind of interaction. But if you keep on pushing this atom harder and harder against the other atom, as is shown in this figure, so if you keep on decreasing the distance between these two atoms, the blue and the red, then at a time it will happen so that this negative charge of this atom, this negative charge of this atom will start actually reacting or interacting almost or hitting almost the nucleus of the next atom. If that happens, then all of a sudden this van der Waals interaction will be broken, and these two cells will be repelled, these two atoms will be repelled, because they have approached too close to each other. So, there has to be certain distance between these two atoms to form any kind of van der Waals interaction, and that is what is called as van der Waals radius. So, what is the distance that is required for the formation of these non-covalent bonds? That is called the van der Waals radius, and this van der Waals radius is more or less the distance between the outermost electron and the center or the nucleus of the atom. So, when the van der Waals attraction between two atoms exactly balances the repulsion between these two atoms or the electron clouds, the atoms are said to be in van der Waals contact distance. What are the properties? So, for a van der Waals interaction to happen, the internuclear distance is approximately the sum of the corresponding radii of the two participating atoms, as is shown in this. The energy of van der Waals interaction is about 1 kilocalorie per mole, which is again equivalent to the energy that is formed in hydrogen bonds due to hydrogen bonds between two interacting macromolecules like proteins, and it is only slightly higher than the average thermal energy of molecules at 25 degrees centigrade, which is about 0.6 kilocalories per mole. So, van der Waals interaction as well as other non-covalent bonds mediate the binding of many enzymes with their specific substrates. Now, I will move on to a third category of non-covalent interaction called hydrophobic interaction. So, I just now told you so much about something called polarity, polar molecule and so on and so forth. Essentially, polarity or polar molecule is to deal with the charge. So, whenever there is a charge disbalance, one part of a molecule is more charged, negatively charged or positively charged, in whichever form you can think of, then that molecule is called polar. So, if there is a polar molecule, then whatever be the charge, that is capable of interacting with an opposite charge. So, if one part of me is more negative, then this portion is capable of interacting with a more positively charged component. If this portion is more positive, then this is capable of interacting with a more negatively charged component. Therefore, if I am polar, I am capable of interacting heavily with the oppositely charged component. But if I am non-polar, that means there is no difference in the charge component between my sides, and I am as a complete system, I have no distribution of these components, then it becomes non-polar. And non-polar molecules do not contain ions, they possess a dipole moment or become hydrated, because such molecules are insoluble in most of the insoluble in water. When they become insoluble in water, that is where we start calling them hydrophobic. Hydro means water, phobic means you have phobia or you have fear to water. So, you become fearful to water. So, non-polar molecules, because they are non-polar, they have no charge distribution, they cannot interact with water, they cannot become soluble in water, therefore, they are called hydrophobic molecules or non-polar molecules. So, these two terms are very interchangeably used. You got to familiarize yourself by saying non-polar molecules is equivalent to hydrophobic molecules. Hydrophobic and non-polar, they are essentially the same. Contrary, when you say hydrophilic, it is polar. So, hydrophilic, philia means attraction. So, when you are hydro, water, attractive, then hydrophilic. Hydrophilic is like you have likeliness for water, and therefore, those are polar molecules. Hydrophilic are polar, hydrophobic are non-polar. So, for example, the covalent bonds between two carbon atoms, CC bond and CH bond, are almost always non-polar, and in biological system these bonds are plenty. Therefore, these are non-polar. If they are non-polar, that means they are not soluble in water. So, hydrocarbon molecules, which are made up of only hydrogen and carbon, are essentially insoluble in water. I will show you some examples. So, here are some examples and the importance. So, if you look at this molecule, this is a fat molecule or a fatty acid called stearic acid. In this stearic acid, as you can see, there are CH and CC bond, and there are plenty of these in this molecule. So, this is one molecule from here all the way up to here, this is the one molecule. What it consists of is a glycerol. This light blue portion is glycerol and the yellow portion is what is called as stearic acid part. So, this one plus COOH was stearic acid, and CH2OH, CH2OH and CH3OH was the glycerol part. So, glycerol and stearic acid interact to form stearic acid, fatty acid called stearic acid. So, if they form like that, then this part remains hydrophilic, because you remember there is this oxygen atom, and this oxygen atom is electronegative, and therefore, they can actually interact with water, and there is a polarity, and if there is a polarity, they can interact. So, this blue portion is polar, whereas this portion is non-polar. So, this does not like to interact with water, yellow portion, and therefore, it is hydrophobic, and this portion likes to interact with water, therefore, this is hydrophilic. If you have such kind of molecules which is hydrophilic some part and hydrophobic in another part, then this can interact with water, this do not like to interact with water, and if you just place this as a head group with a ball, replace that with a ball or a circle or a sphere like this, and these yellow portions are centered at the center like this, then you can imagine that each one of these threads are one of these chains. So, you can have 2 or 3 threads, and then you can actually form 2 layers, which is what the biological surfaces are. So, all the cells have a bilayer membrane, which is essentially representing this same thing, that is, you have a hydrophilic part which is towards the outside, you have a hydrophilic part towards the inside of the cell, but in the center you have this lipidic layer which is hydrophobic in nature. And when this forms a circle or form a sphere like this is what a cell is, you know. So, these are the membranes. So, these components are always there. As soon as you add water to these kind of amphipathic molecules, where you have both hydrophilic and hydrophobic components, these are called amphipathic molecules. You know, you remember what is amphibian, you know, the frog which can live in water as well as on the surface. So, these are amphipathic molecules similar to that. So, as soon as you add water to these amphipathic molecules, the hydrophilic portions will try to interact with water, whereas the hydrophobic portion, that is these yellow parts, will try to hide away from water and sequester themselves toward the center. No, I do not want to interact with water, and that is what results in the formation of the covering of the cell or the cell by itself. So, you know, when any interaction happens, as I told you right at the beginning, for example, you have one protein here interacting with another protein here. These interactions can happen because of all the 4 different kinds of non-covalent forces or interactions that I just now told you. For example, here you have hydrogen bond formation or some ionic bond formation or hydrophobic interactions or van der Waals interactions or ionic bond formation. So, there are large number of, you know, bonds that are formed between one protein molecule to another protein molecule. Each one of these bonds by itself are very, very weak and feeble in nature, but because there are so many of them that are formed, that is a result why, you know, 2 molecules can interact. So, with that, I sincerely acknowledge the sources of all the information, whether pictorial or any other form, for representation in this lecture series, and I thank particularly this molecular biology book, chapter 2, and I thank you all for your attention. Thank you very much.